Our presentation is on the transformation of E. coli with a green fluorescent protein plasmid by Jessica, Oscar, and Rosa. For the results, we have two control plates that do not have PFG DNA. One of the plates contains E. coli cells, and the other plate contains ampicillin with E. coli cells. We also have two transformation plates that have DNA. One of the plates contains ampicillin with E. coli cells and PFG DNA. The other plate also contains ampicillin with E. coli cells, PFG DNA, plus IPTG. Control plate containing E. coli cells and no PFG DNA resulted in no visible colony formation. However, there was a visible opaque layer of bacterial cells across the plate. This result supports our hypothesis of having E. coli cell growth due to the LB food source and because there was no ampicillin to kill the bacteria off. Since this is a control plate, there was no expected transformation because no PFG DNA was added. It is necessary to run this control plate to make sure that the E. coli cells we are using survive under normal petri dish environment with a food source. Control plate containing ampicillin E. coli cells and no PFG DNA resulted in no formation of colonies. This result supports our hypothesis of having no E. coli cell growth because ampicillin killed the bacteria off. Since this is a control plate with no added PFG DNA, there was no expected transformation. It is necessary to run this control plate to make sure that the E. coli that we are using does not survive in a normal ampicillin environment. Transformation plate containing ampicillin with E. coli cells and PFG DNA resulted in formation of approximately 30 bacterial colonies. The bacteria looks white and did not glow under UV light. This result supports our hypothesis of having a plate with only transformed bacteria surviving. The transformed cells survived because they took in the plasmid with PFG DNA that also has the ampicillin-resistant gene. This plate is important because it allows us to eliminate the non-transformed bacteria. So if we wanted to use these transformed E. coli cells in further experiments, this is a good way to grow them. The reason why the colonies did not glow under UV light was because these cells were not exposed to IPTG. The IPTG is what allows the green fluorescent protein to be expressed. Transformation plate containing ampicillin with E. coli cells, PFG DNA, and IPTG resulted in a formation of approximately 50 bacterial colonies. The bacteria looks white, and they did glow under UV light. This result supports our hypothesis of having a plate with only transformed bacteria surviving. Again, the transformed cells survived because they took in the plasmid with PFG DNA that also has the ampicillin-resistant gene. This result also supports our hypothesis of having the colonies glow under UV light. They glow because the plate was exposed to IPTG and allowed the E. coli cells to express the fluorescent protein. Overall, there was approximately 1,200 number of transformants per microgram of DNA on the plate containing ampicillin E. coli cells and PFG DNA. Also, there was approximately 2,000 number of transformants per microgram of DNA on the plate containing ampicillin E. coli cells, PFG DNA, and IPTG.